So our course name is Advanced Enzymology. Before that, you should know what is enzyme, how this enzyme is useful in our day-to-day life as well as in animal, plant, and all process product. What is enzyme? So enzyme is a biocatalyst. Most of the enzymes are made up of albuminous in nature. So albuminous in nature, so as you know that the protein is classified based on the solubility, albumin, globulin, luteinins and prolamins. So albumin fractions that includes in most of the enzymes. So enzymes that work on specific substrate. So enzyme plus substrate that will convert into a enzyme substrate complex. These enzyme substrate complex finally, it converts into a product, but it requires a certain site of conjunction jointing. So every enzymes are made up for every substrate to convert into a product. So there are a certain enzymologists who has given, for example, Emil Fischer's, they gives a Michaelis Menten uh, this enzyme plus substrate complex, ES complex, that substrate binding with enzymes is affected by many factors. So one of the component that we will already understand what are the enzyme substrate concentration, how it is factor affecting enzyme activity. So pH, temperature, as well as that we will study in the biochemistry, introductory biochemistry. So enzyme is basically classified into a six part. One, EC1, EC2, EC3, EC4, EC5, EC6. Based on oxidoreductase, OTHLIL, oxidoreductase, transferase, lyase, isomerase, ligases. Okay. Likewise, in this course, you have to understand enzymology and enzyme engineering. So this course includes characteristic features of enzymes. What are the characteristics you will find in any small enzymes? As we know that it speed up the reactions, speed up the process, but it requires in very small amount. A small amount of enzyme, when it is added into a reaction, it can proceed or speed up the reaction. Now another characteristic, they speed up the reaction process, but they do not undergo any chemical changes during the complete reactions. It means it is not involved in that. Simply their presence is required. Another things, enzyme get affected by temperature, pH, substrate concentration. Thereby here the enzyme affected by the temperature, which means they can degenerate at high or lower temperature. It means it require efficiently work at optimum temperature. Similarly, optimum pH. Enzymes are also pH sensitive. So it, which means highly acidic or highly basic nature is not suitable for the enzymes. That you have to remember. Highly acidic or highly basic not suitable for the enzyme to survive and perform efficiently. Now another things that I will teach you. How many types of inhibitor, competitive inhibitor, incompetitive inhibitor. So inhibitor are another factor that affects the enzyme activity. Specifically, it requires for the chemical reactions. Enzyme cannot change the free energy of the reaction. Remember these words. Enzyme cannot change the free energy of the reaction, which means they do not change whether the reaction is absorbing energy or releasing energy, or you can say overall energy. But in certain cases, when it works as a protein, that make a difference. Enzymes are critically for every aspect of our life, cellular life enzymes. So one by one we will understand how this is affected, or what are the critical for every aspect of cellular life in the enzyme, it is enzyme, or specifically for works. So if you see the shape and motility of any cells, that is also depend on, or it is the structure of the enzyme which will specify the cell shape and motility too. Certain are enzyme is attached with the acceptor, receptor. So here you will find surface receptors. It has a chemistry with the substrate concentration, with structure also. Any damn reaction cannot be possible without the enzymes. If you want to grow, if you want to develop, if you want to change anything in the life, that can only be possible with the enzyme. For example, here cell cycle. You know where PM80, prophase, metaphase, anaphase, telophase, how these phases is occurring. It requires certain enzymes. So cell cycle is used. Similarly, the concept of metabolism. In that, you will find anabolism plus catabolism. So for anabolism or synthesis of any two compounds and making a one more, that also require enzyme. For example, fructose plus glucose, that will give you a sucrose. So which enzyme is responsible? You will certainly say hydrolytic enzyme. No. Sucrase is there. Invertase is there. Which enzyme? So it has a functions anabolism and catabolism. So every enzyme is not work two-way direction. Sometimes it's only work on one-way direction also. So it also called as reversible enzyme reactions. Okay. For example, in glycolysis and gluconeogenesis, reverse is that, but the enzyme is different. So regulatory enzyme is also that. For transcription, now these were transcriptions you know very well. That DNA to mRNA, mRNA to protein. So DNA to mRNA, these reaction can only be possible with the transcription reactions. Similarly, hormone release. Plant hormones is differ. As you know that they are called as plant growth regulators, PGRs. If we see in the human bodies, they are responsible for various phases of life. Hormones are there. Follicle stimulating hormone that is found in the most of the women. Hmm? Similarly, hormone releases also require enzymes. Any muscles contractions or increasing in the muscles, that is also require enzymes. Every metabolic activity, whether it is carbohydrate, protein, lipid, fat, fatty acid synthesis, wherever you will find synthesis, whether for example protein synthesis, that also require enzymes. Joining of two amino acids joined together by peptide bonds, it requires also, whether it is proteosis. So in that cases, now we will only understand how it is useful or specific to specific cycles or specific reaction. Now we will understand whether it has any unit. Yes or no? Yes, it has a specific unit. Whether it is in kettle form, whether it is in international union, who has given this enzyme? Whether it is in specific activity, milligram per gram fresh weight per protein, per gram protein, or it is the amount that catalyzes the formation of, remember these words, formation of 1 micromole of product per minute. Standard enzyme unit is an amount that catalyze the formation of 1 micromole of product per minute under defined assay condition. That you have to remember in inverted comma, under defined assay conditions. The unit has the dimension of micromole per minute, one of the unit. Now, the more number of units in a sample means more enzyme catalytes present in that sample. While 1 unit of enzyme is a standard assay procedure, micromoles of the product per minute. 2 unit of same enzyme that gives 2 micromole per minute of the product. So the enzyme concentration in a given sample is generally expressed in terms of unit per ml. So enzyme activity unit may be expressed in terms of micromole substrate per minute, micromole substrate consumed per minute. For any reaction with the defined substrate product, stoichiometry, this can be converted into a standard unit. So according to the unit, you can conveniently by suitable changing their unit according to the product form. Micromole, nanomole, millimole, all you can get per minute, per hour, okay, per second. For example, peroxidase, delta change per minute per gram fresh weight. PPO, polyphenol oxidase, delta change, OD change per minute per gram fresh weight per unit of protein. You can like this specific activity. So ketal corresponding to the enzyme that produce 1 moles of product per second. That is called ketal. Now, before understanding this, we should understand uh, accurate things. Who, what are the scientists? How many scientists involved in enzymology? Or to study the enzymes. Without history, you cannot understand who is the pioneer in the enzymes. So we will start with the 17th century, 1751, up to 2023. First in birds, gastric digestion in the birds. That was discovered or you can published in 1751 by Reumer. Then after 5 years, digestion of meat by gastric juice was invented by this Spallanzani. Then amylase first digest activity. For example, amylase you will find every starch is degraded or amylose and amylopectin. Similarly, amylase activity after 1833. Now remember how many gaps found in the enzymological study. So Berzelius, in many books you will find or I can say it started in 1833. Then after every books are written on the enzyme chapter with after 1836 because Berzelius accepted this concept, catalysis concept. So Kuhne gives these term enzyme term. Until that, no one knows what is enzymes. They are understanding only the reaction is required. This. So Takamine patent on fungal diastase first in 1894. Similarly, same year, 1894, Emil Fischer, that gives a lock and key concept. They describe that every key is related or specific to every lock. So, Bertrand coenzyme, then duck locks, they gives, remember this scientist, 1898, enzyme named to end with suffix ASE. Then Henry, hyperbolic rate of equations. Then Sorensen, who has given 1909 pH scale and buffers. It is relevant to that, therefore I have given here. Sorensen's SPL Sorensen's 1909 pH scale and buffers. Now the very important concept for your advanced enzymology, you have to remember these scientists, Michaelis and M. Maintain equations, equilibrium of treatments for ES complex given in 1930. So we'll start that theory of enzyme actions in 1922. Sumner, remember this is also important, in many books you will find this enzyme urease. He has purified and crystallized these enzymes, Sumner. Then another important enzymologist, or you can say double reciprocal plot, that is in 1934, given by Lineweaver and D. Burke. So Stone first ES complex observed in 1935. Then Dowdrop radioisotope used in the enzyme mechanism. Augustin in asymmetric interaction with the substrate. Pauling enzyme binds. Then Westimor, that is enzyme hydride transfer. Now another scientist you have to remember, that is Costland. D. Costland Jr. In 1958, they have given a hypothesis of induced fit hypothesis in the enzymes. Now, CHW. CHW Hirsch and his co-workers studied on 1960 first enzyme sequence. First enzyme sequence RNase A. In 1961, enzyme commissions was formed and enzyme classification and nomenclature was given by this commission. EC1, EC2 and 1.12, that is given by. After Phillips and its co-worker, first enzyme structure, that enzyme was lysosomes, or you can say lysozyme. So in 1963, Cleland systematization of enzyme kinetic studies was did or happened. Then after models of allostatic transitions given by Jacob and Monod and his co-workers. Then same enzyme after CHW Hirsch and his co-workers, RNase A chemically synthesized by R.B. Merrifield. Then Altman and Zach catalysis of RNA molecules. So Nobel laureates we should know. When we are studying with the enzymology or enzyme engineering, or you can say advanced enzymology, you should know which scientist got Nobel Prize or Nobel winners in this science. So enzymology it will start with the 19th century, 1902, the first Emil Fischer stoichiometrics, or you can say stereochemistry and lock and key concept was given by this Fischer, and he was awarded Nobel laureates. Arrhenius, activation energy and catalysis. Then Buckner in 1907, cell-free extract and fermentation. Remember this E. Buckner. It, he is a very important for the fermentation product. Then I Jackman and F.G. Hopkins. Now, Frederick Hopkins is the father of biochemistry, Sir Frederick Hopkins. Here is F.G. Hopkins. In 1929, remember these words, 1929, he was Nobel laureate for the three things: vitamins, nutrition, and its also contribution in coenzymes. Now, Warburg, remember this, respiratory enzymes. No one will tell you what are the respiratory enzymes. But in past, if you read what are the respiratory enzymes and how it's work, this scientist is very important to understand the COVID-19 also. Okay. So Zant and Gyrogee catalysis of TCA cycle was given in 1937. This is also Nobel laureates. So Kuhne, Fleming, Kunitz, Somnar, Norsopse, Corey and Corey, that enzymes of glycogen metabolisms. This Pauling, Sanger's, lastly insulin sequencing through proteases in 1958 and 1960. This nucleic acid biosynthesis enzymes was given by Okoa and Korenbergs. So likewise, the scientist up to 2018. So structure of vitamin B12 given by, tell me, Crowfoot and Hodgkins, or you silently Hodgkins. Similarly, DNA sequences DD-NTP method with enzymes. Who has given? And it is also a Nobel laureate. F. Sanger. Okay, Sanger sequences. Altman and Sack, catalysis by RNA molecules. Okay. Similarly, Arnold, F.H. Arnold, direct evolution of enzyme or directed evolution of enzyme. So there are Nobel laureates like Kerry Mullis, we are also including this in biotechnology and our course also polymerase chain re- reactions. So now the question arises. We have started with the introductory parts of enzyme classification, enzyme functions, enzyme characteristic, then history of enzyme, what are the Nobel laureates. Now I am starting with which are the techniques, how we will determine. Foremost important, up to these, many scientists are working only in the spectroscopic method. Spectroscopic method. UV visible spectroscopy is most prevalent instrument used for detection of enzyme activities. Changes in the OD or changes in the product formations by which they are understanding. So for example, enzyme say like alcohol dehydrogenase, lactate dehydrogenase or malate dehydrogenase. The detection of NADH, that is at 340 nanometers in UV range. Similarly, visible spectrophotometers, alkaline phosphatase, very important medical enzymes. These alkaline phosphatase can also be determined whether you have drunken or not drunken, or your kidney function is very good or not good or appropriate. This then invertase enzymes that can also be detected through polarimeters or polarimetry, optical rotation, how the degree changes. Turbidometry, that is detection through the attenuation of incident light, scattered light. So you can measure the lysozyme with that turbidometry. Nephelometry. Then fluorometry. It is the also technique through which whether it is decreasing or increasing at 470 or 510 nanometers. So these are the chymotrypsin or cholinesterase is the enzyme. Similarly, luciferin at delta change 562 nanometers, you can measure luciferase in luminometry. Luminometry. So these are the instruments you have to remember and the principle and how it's works in the enzymology. Okay, that one can ask also which are the techniques useful for the measurement of enzymes or enzyme assay. Then another method through which you can also measure the enzyme activity, that is electrochemical measurement. So how detection of hydrogen changes, H plus changes, that you know very well, pH meter for determination of pH increasing or decreasing, the changes in the urease or glucose oxidase or lipase activity can also be measured with that. Generally, lipase activity in rancidity, rancid crop, for example, bajra, okay, lipase activity is very important. So through the instrument of pH meter, you can measure the lipase activity. Carbon dioxide carbometer, likewise potentiometer, amperometer, radio tracer measurement. Yes, it is also used as the detection of beta emissions with this enzyme. Or you can measure the metabolic activities or metabolism one substrate to the another substrate. Pathways analysis then can also be possible with this scintillation counter. Okay, that. These are the techniques through which you can measure the enzyme activities. So up to that, we will understand the fundamental things, how the enzyme is formed and what are the